directly move to the next session, session 9 on aging shifts biology 1. And I'm Hong Gil-lam from South Korea, and I'm co-chairing this session with Yushin Sir. And our first speaker is Frank Slack. He'll be speaking about microRNAs in aging.
Would you turn that computer on? Okay, fantastic. Well, I'll begin first by thanking the organizers for the opportunity to attend this meeting and to present our research. Today I'm going to tell you about a topic that's just been touched on briefly at this meeting so far, but one which I think has huge implications for all the phenomena that we've heard about at this meeting, and potentially touches on all of the genes, all of your favorite genes that we've heard about at this meeting as well, and that is the regulation of gene expression by non-coding RNAs. Now, microRNAs have led the charge in the non-coding RNA revolution, and for that reason, I'll focus exclusively on microRNAs today. However, I'm pretty certain that if you come to this meeting in a year or two, you'll be hearing about non-coding RNAs, other than microRNAs, also playing roles in aging. But we think that microRNAs and other non-coding RNAs are so important in our development and in our homeostasis that at the new Institute for RNA Medicine at Harvard Medical School, we're trying to harness some of the basic discoveries in this new area of science, these non-coding RNAs, as new medicines to treat some of the great diseases of our time. And as you can appreciate, we've begun by looking at the diseases of aging, including cancer, cardiovascular disease, metabolic disease, and neurological disease. And we think that non-coding RNAs are not only going to be useful as new biomarkers or new diagnostics, but also useful as new therapeutics. I won't have time to talk about our efforts in really translating those discoveries today, but I'm going to tell you more about the basic science of understanding microRNAs and their roles in aging. So a little bit about microRNAs to begin with. I should mention that they're actually small non-coding RNAs of about 22 nucleotides in length, but they get transcribed from our very own genomes. In fact, the human genome makes upwards of 2,500 microRNAs, and we still understand the function of just a handful of those RNAs. So as you can appreciate, there's a lot of functional work that still needs to be done in trying to sift through all of this new data, this really exciting new science of non-coding RNAs. But these RNAs are transcribed from our genomes as slightly longer RNAs. They get processed in a series of steps, including hydrolysis by Drosha and DICER, two of the famous RNases that you've probably heard about in this particular field, ultimately ending up as a 22 nucleotide single-stranded RNA. And this RNA makes its way into a protein complex called the RNA-induced silencing complex, or RISC, where it acts to guide RISC to partially complementary sequences and downstream target messenger RNAs, typically messenger RNAs, although we now know that non-coding RNAs combine non-coding RNAs, and there's a whole new emerging area of non-coding RNA binding to itself and other classes of non-coding RNAs as well. And when that happens, RISC functions to shut off the activity or the expression of these particular RNAs. And typically that manifests itself as a reduction in the levels of the messenger RNA that is being targeted by a microRNA. So one of the most important components of RISC is a protein called Argonaute. In C. elegans, it's known as ALG1, and it'll be the star of the show in my talk today. Argonaute is, in fact, the protein that directly binds to these small RNAs, and in some cases has the cleavage activity in cases where a microRNA can cause cleavage of a downstream target RNA. I should say this is very rare in animals, but it does occur quite frequently in plants. Maybe we'll hear more about that at the meeting later on. So I'm not going to have time to go into a lot of background about the roles of microRNAs in aging. I'll give you a few pieces of data from my lab and from the field, but it's basically going to revolve around this. We know that microRNAs are intricately involved in aging. We know that because they're required for normal aging. I'm going to give you some examples from C. elegans, but there are examples from fruit flies as well of cases where, in the absence of a microRNA gene, the animal or the population of animals has an altered lifespan. We know that microRNAs frequently change their expression during aging. In fact, there's a generally accepted view that over the course of aging, microRNA levels tend to decrease as animals age. And I'll try to address why we think that happens during this talk today. And thirdly, microRNAs regulate genes that are known to function in aging pathways. I'll give you some examples of microRNAs that are regulating the insulin signaling pathway, for example, today. But almost all of the genes that I've heard about this meeting, I can guarantee you there are microRNAs that are binding to the 3' UTRs of those genes and causing altered expression of those genes. So something to keep in mind when you're expressing your favorite gene and you want to know what it's truly doing, make sure you keep its 3' UTR on the construct so you don't lose the control by microRNAs. All right, so we came to this field of aging as developmental biologists. I'm a developmental biologist. I think of development as a stage of I think of aging as a stage of development. It's like the end game of development. And so we use the logic and the tools of developmental biology and developmental genetics to try and understand aging. And so for many years, my lab has studied the role of microRNAs in the timing of stem cell development in C. elegans. And two of the most famous microRNAs involved in that process are LIN4 and LED7, which begin to be expressed at particular times during the larval stages of C. elegans and control what fate stem cells are going to adopt at those particular times. So we asked a simple question to begin with. The first was, if microRNAs can control the timing of development, can they also control the timing of aging? This was sort of a heretical question to ask to a certain extent, given some of the objections to the thought that aging could be programmed. But I'm going to give you some examples that we believe that there's a smidgen of evidence to support that idea. And the second question that we wanted to know was, well, if certain microRNAs are coming on at these times during larval stages, are there other microRNAs that are coming on at later stages, maybe in early adulthood or during adulthood, that could be controlling other switches and maybe controlling the timing of aging? So to answer the first question, we just asked what would happen if you knocked out some of those microRNAs, like LIN4. And this was a sort of a surprising study, but back in 2005, it really led to the first evidence that microRNAs could play roles in aging. And that was that if you knock out LIN4, these animals, compared to a population of wild-type animals, have a very short lifespan. And this wasn't just because these animals were sick. Rather, we could see that these animals precociously expressed markers of old animals, even though they were sort of young in adulthood. And secondly, if we overexpressed LIN4 just from a multi-copy plasmid, we could see that the animals had an extended lifespan. So it really said that these RNAs were actively controlling the longevity of this population of C. elegans. We also showed a few years later that if you knock out the Argonaute protein, this is ALG1, sorry, the gene for the Argonaute protein called ALG1 in C. elegans, and you knock it out just during adulthood, so you can grow the animals on normal OP50, which is the normal E. coli strain that we grow them on, and then at day 0, when the animals have just molted from the larval stage 4 into adulthood, you grow them now on RNAi against the ALG1 gene. We saw that these animals have a slightly shorter lifespan as a population. That says that ALG1 is, something about ALG1 is necessary for normal lifespan in C. elegans. And one of the features of ALG1 that's not really appreciated, people think of ALG1 as the sort of functional unit of risk, but in fact, Argonaute has other functions, including stabilizing the microRNAs that are within risk. There's some evidence that microRNAs within risk are stable even in our blood. So Argonaute has a very stabilizing activity on RNA levels. And what we were able to show is that as C. elegans age, there are certain microRNAs that get upregulated during aging. I'll talk more about some of these today. But in the ALG1 loss of function, when you would just knock it out in the adult stage, we see reduced levels of these RNAs. And that's presumably because ALG1 is required for stabilizing these RNAs. In addition, there's some evidence that ALG1 might be involved in the processing reaction of the steps of maturing microRNAs from the hairpin loop structure. So ALG1 might also be involved in expression of microRNAs. So in 2006, Monica Driscoll's lab showed for the first time that there are many microRNAs that change their expression as C. elegans age. And this is now a common feature in every case that I've seen in the literature. Maybe I haven't seen the negative result papers, but in every case that's been published in other animal species, and we've done this ourselves in mice as they age, you can see changes in the levels of microRNAs. And in some cases, microRNAs become increased as animals age, and in other cases, and broadly this is what people see, the vast majority of microRNAs get downregulated as animals age. So when we were looking for this sort of hallelujah microRNA that might be controlling the timing of aging, we wondered if it sat in some of these examples here where the microRNAs were expressed at high levels at particular times in adulthood. And one that I'll focus on quite a lot today because I think it illustrates very nicely the power of these tiny little genes to control aging processes is this gene, miR71 over here. You can see that Monica's lab showed that very early in adulthood, this microRNA gets upregulated and then it gets shut off again as the animals age. So it sort of peaks at early adulthood. I'm going to tell you that this microRNA is not only required for normal aging, but this peak in the earliest stages of adulthood is predictive of future lifespan of animals. The animals that express more of miR71 here live longer than the animals that express less of it. In addition, this microRNA I'm going to show you today regulates some of the key factors that are involved in aging in C. elegans, including multiple members of the insulin-like signaling pathway. So when high-throughput sequencing became in vogue, we, of course, did some of this first small RNA sequencing on aging animals, and we saw a similar trend to what the Driscoll lab had shown, and that was that if you just look at microRNAs now in wild-type animals shown here in blue, this is a waterfall plot where microRNAs that are most upregulated in aging are shown here on the left, and the ones that are most downregulated in aging are shown on the right. And you just rank them based on their fold change. You can see that for the most part, most microRNAs get downregulated. And this was true even in a DAF2 mutant. So that's the gold over here. We've ranked the microRNAs. We've shown the same microRNAs in gold as the ones next to them in blue, but they're ranked by N2. So some of the microRNAs on this list that we published on before include miR71, which is one of the most upregulated microRNAs, even though it sort of peaks at a very early time in adulthood. One of the most downregulated is LET7 of here, one of my favorite microRNAs, and one of the ones I mentioned earlier that's involved in controlling stem cell fate during C. elegans development. All right, so some of the questions that these new data posed to us were, do additional known microRNAs play roles in aging? And are there, in fact, novel microRNAs that we hadn't yet discovered because they only play a role in aging, and maybe we hadn't profiled out in aging to discover them? What pathways and networks do these microRNAs, these aging-associated microRNAs, function in? And what causes the altered expression changes that are found in aging? What causes those upregulated ones to be upregulated and the downregulated ones to be downregulated? And can these microRNA expression patterns be used as biomarkers of aging? I think I've already given away one of the answers to that question already. And then lastly, are there diagnostic, prognostic, or therapeutic uses for these microRNAs in diseases of aging? I won't talk to you at all about this today, but I'm sure you can appreciate that if we do find factors that are regulating insulin signaling, those could be really useful potentially in metabolic disease. If we find microRNAs that are regulating oncogenes, they could be really useful in treating cancer. And this is the whole premise for our new Institute of RNA Medicine. All right, so just to give you a little bit of background, we published this before. I'm just going to highlight a few examples of microRNAs that look like they're controlling lifespan in a population of C. elegans. You've seen these kinds of curves before. Here's N2, the population with an average lifespan of just over 2 weeks. In the case of miR71 mutants, they have an extremely short lifespan, about half the lifespan of a wild-type strain. Much like what we saw for the case of LIN4, if you overexpress miR71, we could extend lifespan as well. This is one of the sort of gold standards of a real gerontogene in that it's not just causing sickness, it's truly able to give a long or a short lifespan depending on how you manipulate the gene. And in addition, miR71 functions within the insulin signaling pathway. We were able to show that the long lifespan of DAF2 mutants depends on a wild-type copy of miR71. Now, another thing that I want to point out, because this is something that isn't really looked at very much, and we haven't heard much about it at this meeting today, this week, is that what you're looking at here with one of these lifespan curves, these sort of Kaplan-Meier curves, is a population that's, in the case of C. elegans, genetically identical. These are isogenic siblings in many cases, growing under the exact same conditions, often on the same plate, in the same position in the incubator, growing on the same food. And yet some of the animals are dying within a week, and others of them are dying within 3 weeks. So this variation in lifespan is something that's intrigued me. And one thing that we noticed quite quickly when looking at the miR71 mutants is that the variation in lifespan is dramatically shortened or reduced in miR71. You can tell that in this one day when we counted the number of animals that died, more than half the animals died within a single day. So the perfect mutant would have had all the animals alive on one day and all the animals dead on the other day. But this is about as close as we've ever seen. So miR71 is somehow required for this variation in lifespan that's apparent not only in these C. elegans mutants, but of course this variation in lifespan is apparent in every species that's been looked at. And we're wondering whether microRNAs could play a role in that as well. And I'll give you some evidence later on that it indeed is the case. All right, so just quickly, we showed that miR71 regulates multiple members of the insulin-like signaling pathway, including the PI3 kinase AGE1 and the PDK1 kinase. This is some of the evidence for that. I'm not going to go into it because this work has been published. And in addition, miR71 also regulates part of the DNA damage response genes that have been shown to also play roles in controlling lifespan in C. elegans. Additional microRNAs play roles in this pathway as well, although we don't quite understand what those targets are. So without going into any detail, I'm not going to even describe this particular figure to you, but just briefly, we took all of the 80 aging-associated microRNAs that we and others have identified, and we asked, what are the complete set of downstream targets that those microRNAs could potentially regulate just by using target prediction algorithms? And what you can see here is that this set of genes includes almost everything that we've heard about at this meeting. So that's why I think I make the assertion that microRNAs are going to play roles in your favorite pathway. You might want to start looking at it. But they include phagosome, lysosome, spliceosome. These are all things that we've heard about at this particular meeting. All right, so we think we have a handle on what the microRNAs might be regulating downstream, but we still have very little idea about what might be upstream of some of these microRNAs. So what's causing them to change their expression during aging? So a graduate student in my lab, Sachi Inoke, built a network of transcription factors and microRNAs based on data coming from the Modern Code project, which is the encyclopedia of coding DNA elements, of which my lab was a part. If you just ask, well, hopefully you can see something like this, but the most connected genes are shown down here, and then the least connected genes are shown all the way around over here again. Blue just means that a transcription factor is known to bind in the 2 kb upstream of a microRNA promoter, and the pink just shows that a microRNA is strongly predicted based on physical evidence, meaning that we have pull-downs of Argonaute, and we know where the microRNAs are bound in the genome to the 3' UTR of that transcription factor. Some of the things that emerged from this particular study were that the 2 transcription factors that were most connected in this analysis were FAR4 and SKIN1. We've heard a lot about SKIN1 at this meeting, a little less about FAR4, but FAR4 is a homologue of DAF16, which is a FOXO 4-kid transcription factor. And the most connected microRNA was miR71. I've already told you this is a microRNA that we know is required for normal aging. In fact, if you just pass out this network a little bit and ask which of the microRNAs are most connected to FAR4 and SKIN1, it turns out that miR71 and another microRNA, miR228, are both targeting those transcription factors and are bound in their promoters by those two transcription factors. Now you might be wondering, why did we focus on those two? Well, of course, I mean, they were the most connected transcription factors in our network, but secondly, they're also two transcription factors that have been most highly linked to dietary restriction lifespan extension in C. elegans. So we knew about miR71. We knew nothing about miR228. This microRNA, all we knew was that it was upregulated during aging, but we hadn't had the opportunity yet to knock it out and ask what its function may be. What I'm going to show you is that maybe even in the next slide, is that miR228, like miR71, is required for normal aging. And in fact, these two microRNAs play in a feedback loop with FAR4 and SKIN1 and are both required for the dietary restriction-induced longevity seen in C. elegans. So just to give you some of the evidence for that, I'm not going to go through it all because this work has been published just last year. But once again, in a wild-type animal, we see this survival curve. Turns out that miR228 normally antagonizes lifespan. So if you knock out miR228, animals as a population have a longer lifespan. And in fact, this long lifespan depends on wild-type levels of FAR4 and SKIN1, because if you do the double knockouts, you can find that those animals have a short lifespan again. And in fact, miR228 is required for the normal expression of those two transcription factors, much like we predicted based on the fact that there are binding sites for miR228 in the 3' UTRs of these genes. And a similar case was seen for miR71. Looked like it was required to repress FAR4 but activate SKIN1. And in our paper, we have a model that describes additional data, building a sort of, so taking that network analysis into a molecular pathway. Just a few pieces of data. This just shows you that if we take a FAR4 GFP fusion that actually has the 3' UTR of FAR4 on it, we can see that in the absence of miR71, we see dramatically increased expression of FAR4. So just reinforcing what I showed you earlier, that miR71 looks like it indeed regulates FAR4. So we asked whether these two microRNAs might be implicated somehow in DR-induced longevity. The first thing we looked at was just whether these microRNAs change their expression in the presence of DR, and it turns out that they do. If you use an E2 mutant to induce DR in this case, but we've tried other DR-inducing approaches as well, we can see increases in the expression of both of these microRNAs. And then the second question you asked is, were they required for the DR effect of increased longevity? And you can see that while wild-type animals have this average lifespan, if you induce DR in these animals, you can see that you get an extension of lifespan. But this does not happen in a miR71 mutant. Now, the miR228 mutants I've told you already have a slightly longer lifespan, but that lifespan is not further extended by dietary restriction. So we think that miR71 is in fact required for the FAR4, FOX, and SKIN1 dietary restriction extended lifespan. miR228, we're not sure exactly how it's functioning, but one way I think about it is that the miR228 mutants are hallucinating that they're in dietary restriction already, and that they have already sort of maximized the extension that you're going to see by dietary restriction. Okay, so turning now to whether we can use the levels of these microRNAs as a sort of a biomarker, if you will, for future longevity, we asked whether we could monitor the levels of activity of the promoters of some of these key aging-associated microRNAs over aging in individual animals, and then image them over time, and ultimately try and correlate the expression patterns of those microRNAs with the ultimate longevity of each individual animal. And what we'll be able to do in 4 cases, I'm just going to highlight miR71 for you in this case. This is the top left box here, was we saw that for the most part, if you image animals when they're young, they have low levels of activity of the promoter. We've taken the promoter here of miR71 and fused it to GFP. It's a commonly used way of just monitoring the activity or the expression of a gene in C. elegans. What we can find is that if you follow those animals over time, they start to express more of the microRNA, and then ultimately the levels of activity of that promoter reduce over time. Now, this mimics what we had seen already by the microarray and by the genome sequence, by the small RNA sequencing, where we noticed that miR-71 kind of peaked at an early point in adulthood. So just within a few days of the animals becoming adult, they were already expressing the highest levels of miR-71. One other thing that you can appreciate, hopefully, is that some of the animals expressed more, or at least had higher promoter activity than did other animals. And so we wondered whether the animals that were expressing more of the microRNA might have a slightly longer longevity. And in fact, this was indeed what we saw. The animals that expressed very low levels of miR-71 or just turned it on for a small amount of time and turned it off very quickly ultimately had a short lifespan. So this is daily imaging of the same worm, and that worm is shown over here. It sort of has a little bit of miR-71 expression, shuts it off, and ultimately the animal dies. In contrast, animals that express high levels of miR-71 and express it for longer, like shown by this animal over here, have a longer lifespan. So take a moment to think a little bit about this, because this is a biomarker that we're measuring in the very earliest stages of adulthood, about day 1 and day 2 of adulthood, that is ultimately predicting the future lifespan of this particular population. And as far as I know, to date, this is the only strong single gene marker for as a biomarker of aging that you could measure at the very earliest stages of adulthood. And we're taking this idea to the next level by collaborating with Luigi Ferrucci at the NIA and the Baltimore Longitudinal Study of Aging to ask whether blood samples taken from humans when they were young contain microRNAs that might mark which humans might have had a longer lifespan when they ultimately died. I don't have the results to share with you today. But anyway, to conclude so far, I've told you that microRNAs can promote and they can repress or antagonize lifespan. In some cases, they promote. In other cases, a different one might antagonize. We know that many known C. elegans microRNAs as well as mammalian microRNAs change their expression during aging. In general, there's a global downregulation of microRNAs that's observed. And in the next part of the talk, I'm going to tell you about why we think that's the case. We know that multiple microRNAs are required for normal lifespan, required for the lifespan extension of DR. They're required for stress responses. I haven't had time to tell you about that today. And they fit within aging pathways by targeting key genes in these pathways. In addition, novel microRNAs and targets continue to be discovered through these kinds of processes. So it wouldn't surprise me if the number of aging genes dramatically increases when we start to investigate some of these regions in all of our assays. So microRNAs can act as biomarkers of aging. And lastly, I've highlighted miR-71 as a key aging-associated microRNA that's required for normal aging. One of the disappointments of the miR-71 field for me, if you will, is that as far as we know right now, miR-71, in contrast to many other microRNAs in C. elegans, is not conserved in humans. We do not know of a functional or a sequence homologue of miR-71 yet, but it would not surprise me if there was a gene, a microRNA gene like this, that functioned in a similar way within us. And really, hopefully, people in this audience, maybe our lab will discover such a microRNA in the future. So I'm going to turn my attention for the remainder of the talk to tell you a little bit about what we think is responsible for the changes in gene expression that occur during aging. Many of you have noticed in your own studies that typically messenger RNA levels get deregulated as animals age. Protein levels get deregulated as animals age. MicroRNA levels get deregulated. I've already given you some examples of that today. And we wondered whether these microRNAs that we've been studying in aging might play a role in that sort of global downregulation or that global misregulation of molecules that occurs during aging. So one piece of data that got us started along this route was that I didn't point out for you in that network analysis that I showed you earlier. But in fact, the most connected gene in this particular network or this particular system was Argonaute. ALG1 was connected to more transcription factors and to more microRNAs than any other factor that we had in our network. And that doesn't even include the edges that you might want to draw from Argonaute to every microRNA, because we know that Argonaute affects the expression of almost every microRNA. So even without those edges, it was still the most connected gene in the pathway, in the system. And so we wondered whether this, being targeted by so many aging-associated microRNAs, could explain something about the loss of microRNA expression that's typically seen in aging. So not only is Argonaute one of the most connected genes in our network, but in fact by a separate analysis that I'm not even going to try to explain to you because I don't quite understand all of the mathematics behind it, its node is the most influential node of any in our network. If you look at a parameter called betweenness centrality that tries to quantify how important each node is in influencing other nodes in the network, ALG1 sits out as an outlier up here. We've got the highest betweenness centrality score of any gene in our network, way more than just the number of neighbors that it has in the network would have predicted it to have. So we focused on ALG1. It turns out that ALG1 is the most targeted 3' UTR of our network as well. It's got at least 47 microRNA binding sites in its 3' UTR, just for the microRNAs that are aging associated. We haven't even looked at whether it's got microRNA binding sites for other microRNAs as well. The microRNA that targets ALG1 most intensely is miR71. So while these other microRNAs might have an equal or be important in targeting ALG1, we haven't taken it upon ourselves to prove that they indeed regulate ALG1. But we have asked whether miR71, in fact, regulates ALG1. So the first thing that, so let me just point out a few things about the 3' UTR of ALG1. So if you just take a snapshot from the UCSC browser, you can see that the ALG1 coding sequence is highly conserved amongst nematodes. This is where the 3' UTR begins and goes out. It's quite a long 3' UTR. But you can see that many of the miR71 binding sites shown here by these blue and red colors lie within regions that are almost 100% conserved between all the nematode species, suggesting that these are important sites that have been maintained for a reason. And I'm just highlighting these two over here because I'm going to show you a little later on that if we just delete these two sites from the 3' UTR of ALG1, we can see alterations in the expression levels of ALG1. Okay, so just those two sites are, we know are important, but these other sites are probably important as well. We just haven't gone and knocked them all out. That would just be a heroic effort to knock out all 15 of those sites. But just a few things to point out. We noticed early on in sort of gene expression databases that the levels of ALG1 was declining as animals aged. And Sachi was able to validate that by qRT-PCR. You can see that from day 1 to day 3 of adulthood, the levels of ALG1 plunge. Okay. And this is a time when most people thought that C. elegans was happily non-aging. I'm really happy to see at this meeting that there are cognitive declines and other declines that are happening around this time that really suggest that at the beginning of adulthood, these animals are already in decline. All right, so to ask whether miR71 could be responsible for this plunge, Sachi asked, what happens if you knocked out miR71? So in a miR71 mutant, this plunge doesn't happen. So ALG1 levels do not get switched off as animals age in a miR71 mutant. This says that ALG1 is, in fact, highly likely to be regulated by miR71, and that miR71 is at least partially responsible for this decrease in expression of ALG1 over the beginning of aging. She also fused ALG1 to the 3' UTR of ALG1 to a GFP reporter and was able to show that the levels of that reporter gene are also elevated in a miR71 mutant. In addition, the levels of ALG1 protein are also elevated in a miR71 mutant. And lastly, I told you that we knocked out those two highly conserved miR71 sites, and she was able to show again that the levels of ALG1 reporter were also increased if you lack those two sites. So taken together, that really says that miR71 is at least part of the reason why Argonaute levels decline as animals age. Now, one thing that I've mentioned to you is that ALG1 is required to stabilize or even to be involved in the expression of many different microRNAs. And so I've also mentioned that in a miR71 mutant, Argonaute levels remain high even as the animals are aging. So what Sachi next asked was, what happens to just microRNA levels as animals age in a miR-71 mutant? You might predict that with a miR-71 knockout, ALG1 levels would be higher. That would stabilize more microRNAs. And indeed, that's what Sachi was able to show. So if you rank the microRNAs based on their fold change in wild-type animals, shown here in blue, and then superimpose on that the fold change of those same microRNAs in the miR-71 mutant animals, you can see that for the most part, in fact, almost in every case, microRNAs were more highly expressed in aging in a miR-71 mutant than they were in the wild type. There were a few exceptions. Of course, miR-71 is a microRNA that's missing in that mutant, and of course was not detected and isn't shown in this particular waterfall plot. So we validated some of these expression changes by qRT-PCR, and we saw a similar trend, and that was that microRNAs that were often upregulated in aging were even more upregulated in the miR-71 mutant, and some that were downregulated in aging now were downregulated to a less extent in the miR-71 mutant. So if this finding is true, and I've already told you that microRNAs regulate many downstream target genes, many downstream messenger RNA genes, what might happen to downstream mRNA expression in a miR-71 mutant? You can imagine that mRNA levels would be highly dysregulated. And that's, in fact, indeed what Sachi saw. So I'm not going to go into too much detail because my timer just went off. But just to show you the waterfall plot for now for mRNAs that change of aging, this is from 1 to 23,000 different unique messenger RNA genes that are found in C. elegans. You can see that about an equal number of messenger RNAs are upregulated as downregulated as C. elegans age. One thing, and that's sort of shown here by this sort of distribution plot over here. But unlike what we saw for the microRNAs, where more microRNAs are upregulated during aging in the miR-71 mutant, we didn't see a change in the number of genes that were up or down regulated in the number of messenger RNAs that were up and down regulated. But what we saw was quite remarkable. And that was shown here in red, is the fact that these genes were just completely unregulated. Some genes that would normally go up went down, some that would normally go down go up. What was even more remarkable about this was that the error bars for these points, and we weren't able to show it because this is 26,000 points or 25,000 points, the error bars for the red were way tighter than the error bars for the blue. So we did this in 5 different biological replicates. So not only were the mRNAs deregulated, but they were specifically being deregulated. In fact, the variation that we saw, the coefficient of variation that we saw for the messenger RNAs was much reduced in the miR-71 mutant than it was in the wild type. Now, some of you might be aware of this idea that microRNAs might be involved in fine-tuning or involved in reducing noise that one sees in biological systems. And this might be a great example of that. In the miR-71 mutant, we've got increased levels of many microRNAs, and they're therefore decreasing the gene expression noise that one sees in a population of aging animals. So Sachi tried to quantify that as just a sort of a coefficient of variation, and we can see that in the miR-71 mutant, more genes are less noisy than in the wild type as one looks over aging. Now, funny enough, and I've shown you this already, in the miR-71 mutants, we showed that in fact the population of animals itself has a reduced coefficient of variation. So more of the animals will die on the same day than in a wild type population or in animals that overexpress miR-71. Now, we cannot make the direct connection and say that the animals that the reduction in the coefficient of variation in gene expression is directly correlated with the reduction in variation in lifespan, but we'd like to try and make that connection down the road. So just to conclude, I've told you that miR-71 is expressed in early adulthood and it promotes a normal lifespan. Siblings with higher miR-71 in early adulthood will actually live longer. I told you that miR-71 promotes lifespan variation because in a miR-71 mutant, there's actually reduced variation in lifespan. It also promotes gene expression variation. this kind of argues for a directed mechanism of gene expression deregulation, as opposed to the sort of dogma that's out there that gene expression becomes deregulated because of the sort of slings and arrows of old age, the accumulated damage that occurs in animals. So this is a signal that might be coming on in early adulthood that's switching on a program. I say program in inverted commas, so don't hit me too hard for that. A mechanism, a genetic mechanism that's responsible for that program of decline that one sees in normal aging. But it really does beg the question of, does normal lifespan in fact require this gene expression noise? I've told you that miR-71 mutants have less of it and they have a shorter lifespan. So is that gene expression noise something that's been selected for over evolution to allow adaptability in older populations? We'll see. And then lastly, is this microRNA feedback mechanism a common feature of animal aging? miR-71 is not conserved, but of course Argonaute is, and Argonaute we know is targeted by many, many microRNAs in the human genome. And so we would posit that given the conservation of general mechanisms between C. elegans and more complex organisms, that this kind of feedback mechanism where microRNA might regulate Argonaute, which might regulate the expression of microRNAs, could be a common feature of our aging as well. So this is our model that miR-71, I think, regulates Argonaute. It also regulates insulin signaling and the DNA damage response. That leads to changes in expression of other microRNAs, which then leads to changes in expression of target mRNAs, and of course could also lead to gene expression noise, and whether this is a requirement for normal aging waits to be seen. So I'm going to end by thanking you for your attention, and also just thanking the people in my lab who did the work. This was done by mainly 2 graduate students, Sachi and Thaliana, as well as 3 postdocs that have gone on to form their own labs throughout the world. So thanks for your attention.
Thank you. The session is open for the question, comments.
So I'm not going to talk about program aging. I have a bit off-topic question, but the Pandolfi lab showed or suggested that small RNAs were kind of communicating between genes and pseudogenes. So have you ever thought about that in aging? So C. elegans have pseudogenes. Have you looked at that expression with age and then the relevant crosstalk gene to ask if that's actually another mechanism where they're really involving this communication?
Yeah, so that is an excellent question. I would say that the pseudogenes are just one class of RNAs that need to be investigated further. My lab is really just beginning to look at other classes of non-coding RNAs. The pseudogenes are an excellent example of that. There are also long non-coding RNAs. There are circular RNAs, something we haven't even heard about at this meeting. I hope that in 2 years we can come back and tell you that circular RNAs are also involved in aging. But one of the mechanisms for pseudogenes and for circular RNAs is that they sponge up microRNAs, like you mentioned. So finding a sponge for miR-71 would be just the perfect thing, for example, for us to be able to hunt down, to actually show might be an important non-coding RNA. Absolutely something needs to be looked at.
Frank, do you know which microRNAs are required for the DAF2 longevity?
Yeah, so the question was, which microRNAs are required for DAF2 longevity? Well, I showed you one example of miR-71. We also did it for some of the other ones. I just can't remember which ones there are now. We published that on.
Is there a lot or just a few?
Well, we only looked at about 5, so there are many more that we could do. But one reason why we also profiled from the DAF2 mutant was we were hoping we would see some that were just dramatically misregulated.
Right, I see
DAF2.
A number of them changed in the DAF2.
There were a few, and in fact, Sylvia Lee was our collaborator on that project, and that's what she's looking at.
I see.
Which of the microRNAs are involved in DAF2?
Very interesting. You are saying that the Argonaute is to play important roles in the aging of C. elegans. So how about the DGCR8, Deser, and Joser? Is there any evidence in your study?
Yeah. So all of those genes are also downregulated over aging, and all of them have long 3' UTRs that could potentially be targeted by microRNAs. They were in our network, but we didn't focus on them because they didn't leap out the way LG1 did. But I would say almost everything I told you about for LG1 will probably be true for those genes as well. We just haven't investigated it yet.
Thank you.
Yes, very nice talk. miR71 is an important microRNA in aging, I understand it, but my question is, miR71 regulates PHA4 gene only, or it can target other genes or a couple of genes? Because we know microRNA can regulate other genes too.
Oh yeah. Oh yeah, I believe just by our prediction analysis, miR71 regulates, I think, 150 genes. Probably, and that is not probably accurate. There's probably more. So we don't know which are the key ones yet. We haven't knocked them all out in a miR71 mutant and asked which ones are required for normal, for the short lifespan of miR71, for example.
So the other one is very simple question. You showed two genes, PHA4 and SKN1. And is there any physical interaction between these two genes?
Well, there's probably better people in the audience who could answer that question for you, but I can tell you that at least by the modern code data, which is just a ChIP-seq data set, FAR4 and SKN1 sit at many of the same promoters. Now, whether they're physically bound to each other in sort of heterodimers or something like that, that I don't know. Maybe there's somebody else from the Dillon lab here who's working on that. But yeah, they co-regulate many of the same genes, at least at the level of binding to the same promoters.
Thank you.
I think you have shown that Argonaute downregulation decrease lifespan, right?
Yeah.
What about overexpressing it? Does
Yeah.
it increase lifespan?
So one reason why we haven't had a whole lot of luck with that experiment is that Argonaute by itself is actually a developmental gene. So Argonaute's also required for the activity of LET7 and LIN4.
What about just post-reproduction?
Right. So we need to do the experiment in a carefully inducible way. In C. elegans, there are no inducible promoters that don't also in some way affect lifespan. So, I mean, the normal heat shock, which you might do in a yeast, and you can do in a C. elegans if you don't care about aging, you cannot really use that type of system in C. elegans to show that something is involved in aging. So we've been looking quite hard for an inducible promoter that doesn't by itself cause an effect on lifespan.
Given miR71 repress Argonaute
Yes.
and itself extend lifespan, so its major function on lifespan cannot be going through Argonaute.
It's not just through Argonaute. I mean, it's almost certainly has a major role in insulin signaling, for sure. Yeah. Thank you.
Other question? No? Can I ask one question then?
Oh.
So this microRNA is in a sense like kind of mild and like less specific regular gene expression. And do you have some idea on why the animal use these microRNAs for the aging, control of aging?
Well, I don't think that, I mean, even though I may have given you the impression that this is the premier way to regulate aging genes, I really don't feel like that's the case. MicroRNAs, the way I view them, is just a level of gene expression control that is equivalent to transcription factor control. I don't think that you can say that one is more important than the other. And it would take probably a lot of beer for you and I to sit and discuss why nature might have even evolved this kind of method of gene regulation. But I mean, people think that it's a particularly fast way to regulate genes. If you want to hit an RNA that's already expressed, just want to make a tiny RNA that gets transcribed very quickly that can go and hit that particular RNA. This is just hand wave. Thanks.
Okay, that's great talk. Thank you.